Hello and welcome to Lecture 1 for Bio 145 Microbiology. This is the asynchronous or recorded version of the lecture. All right, my name is Jeff Lynn Cheragino, and this lecture is scheduled for Monday, January 12th, 2026. So, as we always do, to begin with at least, we'll start off with the announcements. Welcome to Bio 145. Again, this is for Section 1. My name is Jeff Lynn Cheragino. I'm your lecturer on Mondays, Wednesdays, and Fridays at 11. And this is the recorded lecture that accompanies the live lecture, which we go over in class. Like I said, this is the first asynchronous lecture. It's recorded with narration, and that's why in its title it says show, capital S-H-O-W. So you can always tell that difference from the lectures that I'll be using during the live class. Okay, so this is the show, this is the recorded. To listen to these most efficiently, before doing this, maybe stop right now and read this Word document. It's on Canvas, it's in the lectures folder. It's right at the top. It says Lecture 1, how to watch, edit, blah, 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 Jeff's PowerPoint recorded lectures. All right. And it kind of tells you what's the best way to listen to them. For instance, there's buttons on the far side here, all right, on the corner, but they're kind of wonky to use. So it's better to, if you follow the directions there, more efficiently go through this so you can kind of like add things, change things, type things in, things like that, rewind things easily, stuff like that. All right, so this is a little strange because you should have attended the live synchronous lecture, the live of Monday, January 12th already, okay? This lecture, you're hearing the recording after, but usually the expectation is you'll listen to the recorded one before you come to the live one. A reminder that labs start next week, all right, Tuesday, January 20th, so there's no labs this week. And again, I'm just giving you a suggested reading. Suggested readings, you can see them in the syllabus. They're not required readings. They're just suggested. They'll kind of help reinforce, kind of go over, support the things that I talk about in the recorded lectures and the live lectures. All right. If you want to find it, there's the book, and some people use it, some people don't. Maybe you get a section that the lecture you have a hard time with. Maybe refer to the book. The book is Jeff's Essential Microbiology 2019. It's found inside the book folder, inside the files on Canvas. All right. So you can always find it there. There are some students that use the book intermittently once in a while. There's some students that told me they never use the book. They just use the recorded and live lectures. So it's up to you. So just think about that. Okay. So just to remind you, all the information about the administrative stuff, about the structure or the policies of the course, everything that was in the syllabus, that was covered in the synchronous live lecture of Monday. All right. I'm not going to repeat it in this asynchronous recorded lecture. Okay. Just want that clarify that. All right. From this slide on is going to be testable material. So you have to listen to it, learn it. Some of it I'll go over in the live lectures, and you're free to ask me questions about it, but you're going to be responsible for all of this when test time comes around. Okay. This lecture, it starts slowly. We're just going to do history, and a lot of it's just review for the first few lectures. Review from your just basic biology, introductory biology, 51, 61. But we will accelerate as time goes on. All right, so let's get going.
Figure that out. So the first thing about microbial diversity, okay, when you're talking about microbes, there's a lot of diversity in microbes. Microbes take up a lot of things. So here we have a picture here. I'm going down my pen, by the way. I'm just going to click on my pen. And it kind of shows you some things that might be considered microbes. All right. Got some viruses here. You got some bacteria. But there's other things that are microbes. And they're usually in 5 groups. And these are just layperson's terms. There's bacteria. There's fungi. There's protozoa. There's unicellular algae. And viruses. So those are just 5 basic types of microbes, layperson's terms. Okay, so you should kind of be aware of those 5 groups. Well, what's true of all of them? Well, microbes are organisms that are basically defined by their size. There are organisms that are between, this is true of 99.99% of them, okay, not every one of them, but almost all of them, they're between zero point 0.2 microns in diameter to about 20 microns in diameter. So if you go here, zero point 0.2 is about 200 nanometers. So in this area here, you got your bacteria, your viruses, okay? Mitochondrion is not a living thing, okay? So throw that one out. That's just out there for size. But pretty much in that, from size, defines what a microbe is. It's an organism that between those sizes, all right? It cannot be seen in an unaided eye. That's why you need a microscope to see it, because it's so small. So thinking about these things, bacteria, fungi, protozoans, unicellular algae, viruses, what's true? Are all those eukaryotes or all those prokaryotes, or can they be either? So think about that, what you know about this. If you know, well, yeah, you'd say, well, bacteria are prokaryotes, but fungi are eukaryotes. Protozoans are eukaryotes. So a microbe can be basically either, okay? Either. All right? Either. I'm getting better at this. Okay. Are they unicellular? Are all microbes unicellular? Well, you might think, well, seems like a lot of them are, but the answer is no. Because there are certain, like molds. Molds are fungi. Molds are multicellular. There's some cyanobacteria. Dr. Risser studies. They're multicellular. So microbes can be unicellular or multicellular. What's true is the individual cells are somewhere in this size range, in diameter. That's the key thing about microbes, defined by size. So let's do a little history, because you got to know history for microbiology, at least a little bit. The first person in microbiology history is Anton von Leeuwenhoek. And he was given credit for discovering the microbial world because he came up with the first microscope. Now, he was a Dutch draper, okay? So he bought like materials for drapes. Not the most interesting job in the world, but that's what he did for a living. And to examine the fabric, he needed something to see the fabric. So he created a microscope. And it actually worked pretty good. And, you know, life as a draper wasn't that exciting in the 1600s. And so he started using that microscope to look at other things. And he started seeing things like really microbes, like protozoans, and he called them animalcules. And he saw them in rainwater. He called them scrapings from his teeth. Like I said, life wasn't that interesting back then. And in a lot of things. So with a microscope, he was able to start seeing microbes. Because, like I said, microbes, by definition, are small. Can't see them with your eye. You need something like this. Now you think, wow, microbiology took off then.
And the answer is not really. Von Leeuwenhoek was a very good scientist, and actually, here's a picture of what he saw when he looked in his poop with his microscope. And this is actually a protozoan, and it has to do with diarrheal diseases. And they figured that maybe he had diarrhea, so he decided to look in his poop. And he did quality work, fantastic work, especially with the microscope he had. But its microbiology did not catch on. Why?
Well, two major problems. Number one, he was extremely secretive. He really wouldn't share it. He didn't like to let other people use his microscope. Now, there's a picture of him that he showed like the Queen of England, all right, stuff in his microscope. But he wouldn't let other scientists do it. So do you think people believed him? And the answer is no, because if he wasn't willing to be open about it, and that's the key thing about science, you have to be open, you have to publish, you have to let other people examine it, you know, well, people aren't going to believe it. So that was number one. And number two, basically was this, a lot of people said, okay, let's say you're right, maybe it is this world of microbes that we can't see with our naked eye. But who cares? I mean, most people thought his work was useless. There's no practicality. There's no application to everyday life. That's what people thought at the time. So microbes might be kind of an interesting, trivial thing to most people. So microbiology did not take off in the 1670s or then, all right? Because people couldn't see the practicality. Now, there were certain major events that started to make microbiology look more practical and therefore more important, right? So the first really major one was Louis Pasteur, and we're going to talk about it later because you've probably heard of pasteurization with milk and stuff like that. But really his first major thing for microbiology, okay, he disproved the theory of spontaneous generation. You've probably heard that in 51 and 61. Spontaneous generation said basically dead things could give rise to living things, like rags could become living rats, and soup could suddenly transform into bacteria. Dead things can't become living things. So he disproved that. But his really first big discovery that really made microbiology start to look important, practical, was he showed microbes were used in fermentation. Okay? Because back in his time, water was not safe to drink. But usually alcohols like wine was a lot safer to drink. All right? People drank more alcohol-based things than water-based things. I kid you not. So he first said, okay, he did the study. He showed yeast. He says, hey, yeast, these microbes. He had the microscopes, okay, at this time. He said, yeast can take sugar, grapes, and turn to alcohol and make wine. So he was also, hey, microbes are needed for this process. And people said, hey, that's important because, we drink wine. Wine's important for money. And secondly, he said, well, if you don't have yeast, but instead you have bacteria, they're going to take the sugar and grapes and they're going to turn them not into alcohol, but into acid. Acid is like, think vinegar flavor, okay? That spoils the wine. That would meant the loss of money. So if you saw bacteria, acid, bad wine. Yeast, alcohol, good wine. So he started to show that microbes meant money, all right? Folks, in life, one of the few things I learned in psychology was three things get human beings. Three things are true base motivation, okay? It's like money, sex, and fear, all right? So money was a big deal. So people started to be interested, okay? Then let's talk about the epidemiologist's influence. So epidemiology is a study that hinted, didn't prove, that microbiology was important for understanding disease. So disease, that's another practical application of microbiology. That started to get some interest in. So epidemiology is the study of disease, and it focuses really on three things. And you heard now of the COVID-19 pandemic, you see a lot of epidemiologists being interviewed. It's been a real boost to that profession, okay? So let's say you're studying a disease and you say, I've got a diarrheal disease, okay? It's associated with drinking bacterially contaminated water. So if you drink water that has these bacteria in it, you come down with a disease that causes diarrhea, that leads to diarrhea. So you say the cause, okay? Epidemiology looks at the cause. The cause is the agent that triggers the disease. So in this example, you'd say the cause is some species of bacteria. The source is the holder of the cause. It's the thing that the cause is found in, the cause grows in. In this example, it's dirty, fecally contaminated water. Okay, it's the water is the source. And the mode of transmission, that's the third thing epidemiology. And mode of transmission is just how things get into your body. Here it'd be ingestion, drinking, all right? So that's how epidemiology would apply to this example of this disease. So again, think about cause versus source. Make sure you understand the difference there. Okay, got it? Good. Okay, mode of transmission. Okay, know how that gets into your body, how the microbe gets into the body. All right, so let's look at some key epidemiology things that show that the early microbiologists were epidemiologists, even though they didn't really realize it. Number one, there's a guy named John Snow in England, and he showed people could get sick from drinking certain water that came from a pump. So there was this pump on this river in London, and he said, wow, whenever people drank the water that came out of that pump, they got really sick. And basically it was because human poop, raw sewage, was being dumped right next to the water on the pump, next to the pump. So the water that would be drawn out by the pump had this human sewage in it. Well, big surprise there, okay? And so you'd say the water here is the source, okay? He could say, well, this fecal intake water was the source of this disease. Okay, and he said there's something in this water that's the cause. That was number one. Number two was Ignaz Semmelweis. Okay, he was a medical doctor in Eastern Europe, and he basically worked in a hospital, and he realized that physicians, they could transmit disease to birth mothers, okay, if they didn't wash their hands. And you're like, what? Well, basically, the young doctors, there were student doctors, would work on cadavers in the morning, dead people that had died of some disease recently. And they'd work on them, and then in the afternoon, they'd go down to the maternity ward and deliver babies, all right? And he said, hey, if you don't wash your hands, you're going to transmit disease from those corpses to those birth mothers, all right? And those mothers are going to get sick, and the babies are going to get sick. So he got people to wash their hands. So in this case, the source was, you'd say, well, the source was the dirty hands, all right, the dirty hands. You could also maybe argue that it was the corpses, but that was the holding, whatever the cause was. Now, he didn't know the cause. Like Snow, he really didn't know the cause was a certain type of bacteria. Now, was he respected? Now, you're going to see in my notes, you're going to see a lot of times I'm going to use the word cocktail. What does cocktail mean? Cocktail means this is cocktail party knowledge. Cocktail party knowledge is not something I test on. It's something kind of interesting, trivial, but it's something you could bring up at a cocktail party, okay, hence cocktails, to kind of impress people, to get, conversation going. Let's say you're at a cocktail party and you want to get some icebreaker to start talking to somebody, and so you say, I got this cute little story. Well, these stories I'm giving you is beautiful. You could perfectly use them at a cocktail party to impress someone, or maybe get someone interested in you romantically or something like that. So that's when you see cocktails, think this is a trivial story. It's interesting. It's not going to be tested on, but it's kind of cool and something to know. So in this case, Ignaz Semmelweis, was he respected? I mean, he kind of said, hey, you don't wash your hands, you're going to bring disease to these mothers and babies. And the answer was no, he wasn't respected, because back then they thought washing your hands was something that a wimp would do. It wasn't what a strong man would do. Strong men had dirty hands. And because of that, they made fun of him. And even though he was able to show he could prevent disease by having people wash their hands, the people just said, forget it. And they didn't believe him. And he was kind of tormented and ridiculed, and he ended up dying in an insane asylum. Okay, so sometimes if you're a microbiologist, even though you're right, sometimes you're not going to be respected. All right, something to think about, okay? So, going on now. Now, neither of these people knew what the cause of the disease was. All right, they didn't know, but at least they knew the source. And they started thinking that it might be a microbe. Okay? The actual cause was pathogenic. Pathogenic, think disease-causing bacteria, even though they didn't know it. Okay. Now, this work kind of led to the next stage. And finally, we have a theory. Okay? Should be called a hypothesis, but we call it theory. And that was Richard Bradley. And Richard Bradley was an English botanist, studied plants. And he proposed basically that microbes, here we go, could cause disease. Microbes can cause disease. And that was known as the germ theory. Now, this was earth-shattering because in the 1700s, most contemporary scientists thought disease was caused by physical factors, heat, pressure. And this was the idea of Richard Mead. Okay, Richard Mead was one of those guys who said, oh, it's physical factors. Like, you got a head cold because you washed your hair with cold water. All right? You got a fever because you went into a room that had hot vapors, or it was some salt that you rubbed on your skin. So that was the thinking, okay? Physical factors, pressure, stuff like that. And so Richard Mead and Richard Bradley basically argued one for and one against the germ theory in a bunch of articles, okay, in some very famous medical journals of the 1700s. And basically, people agreed at that time, that Mead was right, that it was physical factors. It wasn't microbes that caused disease. So here again, microbiology does not earn respect. Well, the tide started turning. Again, re-enters Pasteur in the 1800s, and he popularizes the germ theory. He basically says microbes can cause infectious diseases. He says that. And he goes, well, why did he say? He basically said, okay, listen, you take a pot of chicken broth. You got some chicken broth. You leave it open to the air, stock in air. And there's all these microbes in the air, and they're going to fall in the chicken broth, and they're going to start growing there. And if you leave out some chicken broth at room temperature for a few days, it's going to be overgrown by bacteria and, oh gosh, fungi and all a bunch of stuff, okay? Well, basically, he said the same is true if you have a wound, okay? So you have a pot, stuff grows in it. What if you have a wound, a big cut on your skin, you don't do anything, you leave it open in the air? He says, what's going to be the result? And he said, well, what happens? Well, just do it. Well, microbes start to grow in your cut, and you get disease. All right? So that he popularized this germ theory, okay? And a lot of people listened to him because he was Pasteur. He was famous for a lot of things. He ran an institute in Paris. He was funded by Napoleon. They'll have some other great stories about him. And he was big. He was French, and he was famous. But people didn't totally believe him because he could not prove the germ. So, what was Pasteur's problem? Well, he could not get a handle on the microbes. He could not manipulate or isolate them because he could see them under a microscope. He had a microscope, but he was growing them in liquid medium, and he could say, well, look, they're in there. But other people say, well, but in that liquid, there's also a lot of other stuff. You know, there's not just the bacteria or just the other microbe. So he couldn't pick them out. He couldn't purify it. His limitation was he could see them, but he could not isolate them. There was no like purification of the microbes because, you know, he didn't have like tweezers that could pick out that small stuff, and they were floating around the liquid. So this growth on liquid culture was a problem, and it was a limitation. He could not isolate them. He could not purify. Was a guy named Robert Koch. His contemporary was Robert Koch, and he actually proved the germ theory. He was the one. He was able to show, oh, microbes can cause disease, and from then on, microbes were important. Well, how? How could he do that? Well, he established a set of procedures, and they call them Koch's postulates, but they're actually procedures or like steps you would do, okay? And these steps are done to show that a specific microbe causes a disease. And it's used today when they kind of find a new disease and they have to figure out what microbe causes that disease. You'll see in the journal articles, oh, they use Koch's postulates to show the link, to show the cause, the microbe caused this disease. So it's still around today, these steps. Okay. A little cocktail party conversation. Robert Koch was a hick country doctor in what's like Germany, Austria, okay? Pasteur was famous. He had all this money at this institute. Everyone knew Louis Pasteur. He was French. Yeah, But you know, Robert Koch he was poor. He basically worked in a closet in his home, and he studied things like anthrax and tuberculosis, things that could kill him. And they didn't kill him, but he was able to study them, and he was able to come up with the Koch's postulates, the things that prove that a microbe can cause a disease.
So just to sum it up, here is Louis Pasteur working at his Pasteur Institute. Look at all the room he's got, everything. He came up with many, many innovations, but he couldn't prove the germ theory. And here on the right side is Robert Koch, and he's thinking to himself, I figured things out. Why am I working in my closet? But like I said, he was the one who figured out the cause of a lot of these very scary diseases. Very brave guy, too. All right, next slide.
Okay, so what are these Koch's postulates, these procedures, okay? And if we did this live, which I wish we could do, but I'm probably going to do this as a quick kind of hokey demo in class. And a hokey demo is a demonstration where I kind of act things out. And I'll probably do this in the synchronous lecture. Thursday, but let me just kind of talk you through it, okay? You can visualize it. So let's say you used to have a diseased organism, okay, a host organism, Bessie the cow, and she has a disease with very clear symptoms, all right? Very, very clear symptoms, all right? Bessie the cow. That's what you start with. That's number one in Koch's postulates. You say, okay, now I've got to basically isolate, purify that microbe from Bessie's tissues. So imagine there's a bacteria that is the cause of this disease, and it's in the blood. Blood's a tissue. So here's your blood. There you go. It's got a little smiley face and things on it. But imagine again, you got that blood, and remember that blood might have microbes, but it has a lot of other stuff in it too. It has red blood cells. It has the other cells from the human being. It's got salt. It's got warmth. It's got a lot of other things. Okay, you have to isolate the microbe. Things Pasteur couldn't do, Koch could. Well, how do you do that? How did Koch came up? And basically, he used solid nutrient medium. That was his answer. All right. So he could take nutrient medium, like think chicken soup. So you can grow microbes on things like luria broth, tryptic soy broth. These basically soups. They're just soup. But he hardened it. He added agar to it. Agar's like gelatin, jello gelatin. Think about that. He added agar, and it made it hard. It made the medium, the soup, hard. And then he could take this little drop of blood and basically streak it out with a loop, streak that out, spread out everything that's in that loop, in that blood, and wait a few days, and then boom, what would grow here? Pure colonies, right? So these things that are in this drop of blood in a liquid, you see under a microscope. But think about it, folks. Can you pull out something from a drop of blood under a microscope? No, they're too tiny. You don't have tweezers that can do that. So liquid wouldn't be good because it'd be floating around all the time. And liquid things float around. They're tiny. But you put them here, you spread them out with your loop, you streak them out. You'll see this in lab. And now they grow into individual pure colonies. There you go. Can't do that in liquid. Liquid things float around. And then then Koch could then pick up that colony with a sliver, all right, silver, stuff like that, and then inject it into the next host. So he had something isolated, purified. Okay, he could answer Mead's objection. Now what's the next step? You're not done. Well, you take that microbe from this pure culture. We say this is a pure culture now. It's pure. And he inoculated it into a healthy animal. Let's call it Clarabelle. So those bacteria that were in Bessie have been isolated with agar, nutrient, hardened, solidified medium, and injected into Clarabelle. And Clarabelle, if this thing is the cause, Clarabelle should get the same disease symptoms as the original host, Bessie. Okay. All right. Well, that kind of makes sense now, right? Well, are you done? Nope, you're not done. Let's keep going. What do you have to do now? Well, that microbe that you isolated from Bessie, you now have to get it from Clarabelle. So meaning, what does that mean? You have to take Clarabelle's blood. You got to take her blood. You got to streak that out on solidified nutrient medium, hardened, solid. You got to wait, got to grow up find that same agent, that same bacteria, and the same cause, and put it into another animal. We'll call it Bob, okay? Another animal, Bob. And again, if it's true, if you were right, Bob should get the same disease, same symptoms. And for Koch's postulates, you're repeating this over and over many, many times. You have to repeat the above steps many times. You have to constantly isolate the same microbe, which is then going to cause the same symptoms in the new host animals. And then just keep repeating it. Take that blood out, isolate it, inject it, wait for the same symptoms. Then in that animal, take the blood out, isolate it, inject it, wait. And you got to do it multiple times until it's really clear that that microbe, and then only that one, is the cause of the disease. Now, there are other big points in history, but I'm not going to go into them. There's more of them in the online book, but you're not responsible for them. Okay, just make sure you understand the ones I went over. All right, sounds good. Sounds good. Here's just a recap. This is just another take on it. Again, you have a healthy animal, you put the drop of blood, nothing grows, okay? But let's say you get that diseased one, clear symptoms, okay? Very clear, just you take it, you streak it out here, okay? You take it, you can see them, but that does not enough. You got to streak them out, you got to isolate it, you grow it, it's pure now, it's pure culture. You take it, you inject it into a new animal, okay? This is the old one, he's dead. Here's the new one, okay? He's different, he's white. Rabbit, right? Rabbit. I'm not a big-time biologist. I'm a microbiologist. That's a rat. And look, it comes out with the same disease, same symptoms. But that you're not done. You got to, again, take the blood, again, streak it out, isolate one, inject it into another animal. So you got to repeat this multiple times. Okay? That's how you prove the cause of a disease. Koch's postulates. Hope that makes sense to you. Now, we're going to shift gears for the last few slides today, and we're going to start with basic micro concepts. All right, and this should be a review for most of you. But maybe for some of you, you hadn't had this stuff, or it's been a long time. So we're just going to go over it so everyone's on a level playing field. So what's the first basic micro concept? We already kind of said this. Microbes can be prokaryotes or eukaryotes, and some of them are neither. They don't belong to either category. Well, okay, we'll worry about the neither later. So why is that important? Because here's the deal. If you know the category, you can deduce characteristics about that microbe just because you know it's a member of that category. All right, here's my example. Now, I love to make fun of Raider fans. Okay? I'm a big San Francisco 49er fan. And I love to make fun of Raider fans like Dr. Vieira. Okay? So you're like, okay, great, all right. And if you have friends that are Raider fans, you'll find this funny. If you're a Raider fan, you won't like it. But this is all a joke. So let's have a sense of humor, okay? So let's say you find out someone's a Raider fan. Well, because they're a Raider fan, you know there's certain traits that are associated with Raider fans. So I'd say, well, a Raider fan, they're probably, undereducated, they're probably mean, they have a criminal background, ha 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 ha, they like to burn down things. Okay, that's the Raider fan mantra, all right? Trouble, rebel, okay. So the idea there, if you know the category a person's something's a member of, you can guess certain traits. Same thing, if you know a microbe is a prokaryote, you already know certain things about it without even looking at it. If you know it's a eukaryote, you know other certain things are true of it. So if you know the category, you know individual characteristics. So that's, first of all, really important to go over what does it mean to be a prokaryote? What does it mean to be a eukaryote? Okay, because you know that. You know a lot of things about the microbe already. Okay, so first of all, prokaryotes are organisms that have no true nucleus. No true nucleus. So there's domains. So we got to know a little taxonomy for microbes. Not a lot, but so first of all, we're going with the biggest taxon. That's a domain. So we say prokaryotic microbes. They are either the domain Eubacteria or Archaea. Okay. Eukaryotes, on the other hand, have very well-defined membrane-bound nucleus. Okay, and they have a nucleus. Prokaryotes have no nucleus. Now, eukaryotes there's only one domain for you, and that's eukaryota. And what we'll focus on is some of the kingdoms. So in the domain eukaryota, you have fungal kingdom, you have the protist kingdom, and the protist kingdom has unicellular algae and protozoans. So if you go back to the slide we started with, to the 5 main groups, okay, they're bacteria, right? Okay, that's going to be in prokaryotes. But you think, okay, how about fungi, unicellular algae, protozoans? Those are going to be the protist kingdom, okay? Now you say, well, eukaryote also has plants and animals, right? But we don't care. They're not microbes. So for this class, we don't care about plants. We don't care about animals. We're going to focus only on those kingdom, the fungal kingdom and the protist kingdom, because that's where you got your microbes. And of course, the prokaryotic the group, meaning the domains Eubacteria and Archaea. So that's what we're going to focus on, okay? So in terms of that, so here's nice, it kind of sums that. The next picture sums that up. I have to be careful because when I'm switching slides, if I talk too soon, you're going to miss the first thing. So I'm sorry about that. I got to pause. Okay, here's the pause. So here's the domains of the prokaryote. You've got the eubacteria, you got the archaea, and then of the eukaryotes, okay, you just have the eukaryote domain, okay, and the kingdoms we care about are going to be the fungal kingdom and the protist kingdom, okay, and the protists, again, in that kingdom you've got the protozoans, and you got unicellular algae down here, okay, they're down here, all right. So kind of think in terms of boxes now, okay, you've got kind of this drawing, kind of when you're studying, kind of draw this out. You got the prokaryotes, you got the eubacteria domain, you got the archaea domain. The eukaryotes, just one big domain, and you got these different kingdoms, and we're going to care about the fungi, we care about the protist. Okay. Draw things out, always good. Sometimes you're going to hear me say this a million times, the hand remembers better than the brain.
So here's our summary slide of prokaryotes versus eukaryotes in terms of microbes. Prokaryotes, again, are primitive. They have no true nucleus. You're going to when you're talking about them, you're thinking of eubacteria, members of that domain, or we just call them bacteria. So these bacteria, eubacteria. Or you can say these archaea or archae. These are prokaryotic microbes. Okay, use those terms. On the other hand, you can have microbes that are eukaryotes. Eu means true. They have a true nucleus. And you can have fungi and protists. Those are members of kingdoms that are eukaryotic microbes. So that kind of sums it up. Hope that clarifies. Again, draw this out, write this out, do some boxes. Make it easier to remember for yourself.
Now, here I have just a picture, an ooh-ah picture, okay? And it's going to illustrate a certain point. So here are some prokaryotic microbes. You look at them, kind of look like these guys. These kind of look like beads on a string, don't they? Okay. Again, nothing major. Okay. These are okay. These got a little something more there, but pretty basic. These are some eukaryotic microbes. Okay. Any obvious differences? So this is a unicellular alga. Okay. Here's a protozoan that kind of looks like it looks like a dolphin, doesn't it? Or a swordfish. Okay, penicillium. Okay, it looks almost like a flower, doesn't it? Or it looks like a hand from Halloween. Okay, yeah. All right, tetrahymen is another thing. Okay, what would you see just looking at them? What would you say just in general? Major difference. You'd probably just say, just from looking at them, you'd say, hey, the eukaryotes look a lot more complex, right? They're much more fancy. They're much more interesting structures. They're just more complex looking. And that's generally true, okay? Eukaryotic microbes are generally more complex than prokaryotic microbes. So let's do a little thing here, a little activity. You do it at home. You can take a break, stop listening to me for a while, so this is Pete and repeat. There's two pictures, okay? Just do this. You'll see the point in a second. And there's 8 differences between these two pictures. Okay, and I'm just saying find the 8 differences. Okay, and you can stop right now and find the 8 differences. Okay, I'm not going to stop because you don't want me to wait. It'd take me forever. I'm not very good at these. My dad is, but I'm not. So let's just kind of look at, so you can stop it. Do it now. Okay, well, I'm not. I'm going to keep going. Well, number 1, here there's a dog. Here the dog's missing. Okay, that's 1. Okay. This picture has it, this one doesn't. You go other stuff. You go, okay, well, let's look at the back. Look, this looks like a clear day. Here, this looks like the dark. So it's the same, but they're different colors, all right? And there's 8 things, and I'll just let you go through, but you're going to have to painstakingly go back and forth to find the 8 differences, aren't you? Okay? Here's, okay, colored shirt. There's a third one, okay? And you can find the rest of them. Okay? Now, but what's the power of knowing the difference between euks and proks? Okay? What's the power? What's the power? Think about what's the power? So if I say I have a microbe that's a euk, and I say, here's another microbe and a proc, and I say, tell me the differences. Okay, you don't want to be going back and forth and randomly going back and forth, right? Hoping to see the difference. No. If you have two microbes and you say one's a euc and one's a proc, you know what to look for. That's the power. If you know something is a eukaryote, and something, on the other hand, another thing is a prokaryote, you know what to look for because you know the traits, or you should know the traits, that are associated with prokaryotes and eukaryotes. You can see the differences. You're looking for the difference. You know what to look for. That's the power of knowing, oh, a microbe is a prokaryote or a eukaryote. Okay? So that's why you should know what are the basic things about prokaryotes and eukaryotes. All right? Because it can help me predict what a microbe has, a difference one microbe has from another. It's very powerful knowledge. Well, with that in mind, we're going to play kind of a game of same versus different. Okay? Same versus different. And the long-term thing is you want to be able to say what traits are in the eukaryotic box and what traits are in the prokaryotic box. Okay? Because if you know the traits and they're in each box, you can think of, okay, what should I look for? Okay, so think of traits in boxes, and you're going to take out each box. So here's your first proc versus euc. Okay, here's your prokaryote on the left. Here's your big P, prokaryote on the left. Okay, big P. And here is your euc on the right, your basic euc on the right. And right off I can say, oh, this looks a lot more complex, right? This is a lot more complex. It's a lot more stuff. It's a lot more organized. It's, it's really, really complex. This is very primitive, right? This is primitive, all right? So let's kind of go into those. Okay, so let's look here. First of all, what's the same between procs and eucs? Well, what's different? In both proc and euc, you have a cytoplasm. And the cytoplasm is that jelly-like liquid inside the plasma membrane of the cell. So the plasma membrane of a cell usually defined as borders. And inside, everything is called the cytoplasm. It's jelly-like stuff. So that's true of both. Now what's different is eukaryotes are very organized. They have a variety of membrane-bound enclosed compartments. And these membranes have complex metabolic systems. Lots of different proteins with lots of important jobs. And they're like factories. And basically those things, these membrane-bound compartments with these complex metabolic systems, protein workers, these things are called organelles. Organelles are found only in eukaryotes. Prokaryotes do not have organelles. Okay? Prokaryotes do not have organelles. Only eukaryotes have organelles. So that's a big difference. Now, if you have an organelle, okay, inside one organelle, there will be a difference in pH, there will be different protein activities, so forth. All right? So like inside this compartment, all right, are your forks, over here are your spoons, okay? They got different things in them, right? They're separated. They don't get mixed in. So things can be very different here. You can put this water in here if you want to put water in here, and you wouldn't have to worry about water getting over here on your spoons, right? They differ. So in terms of organelles, they differ in pH, protein activities, stuff like that, okay? What's in them? You can think, for instance, a nucleus versus a mitochondria. In a eukaryotic microbe, a nucleus will have a different pH. It'll have different protein activities. It has different contents than a mitochondria, doesn't it? And they're membrane bound, they're separated, so the stuff can't mix together, right? They don't mix together. So each thing can have its own special properties. Special things can happen in one and not the other. You can keep things separate. Think like you have a factory with matches and one with gunpowder. Okay, you don't want them in the same factory. You want them separate. Organelles allow things to stay separate. Okay, and that's really important. And this compartmentalization that you see allows for greater, and we talked about this before when you looked at the two type microbes. The word is complexity. Complexity. There you go. That's the key reason that eucs are much more complex than procs. And because of that, they can do a lot more things. For instance, let's say there's you and your roommate, and you've got everything put away, compartmentalized, everything's straightened up. You got one thing one place, one thing another. You can do a lot of things. Your roommate, maybe in the dorm, is messy. Stuff is everywhere. Everything's mixed together. Their computers, their food, their underwear, everything's all together. They hardly can ever get anything done, right? But you are much more productive. You are more complex. You can do a lot more things. So compartmentalization, organelles, leads to complexity.
So one thing I'd like to emphasize is that prokaryotes don't have true organelles. So remember, organelles is something that's membrane bound, but has like a complex biochemical, biosynthetic pathway inside them. They're like factories. They're basically places where things are taken apart very carefully. They're very organized. They're very complex. All right. Prokaryotes don't have those things. They don't have organelles. Now, prokaryotes can have things called inclusions, granules, bodies. They'll have those. They'll have starch inclusions, starch granules, starch bodies. But these are not truly organelles. Now, what's confusing is they may be these inclusions, granules, or bodies. They can be membrane bound. They have a membrane around them, but they are not truly organelles because they don't have like a complex system of construction or destruction. All right, they're just like warehouses. They're not factories. So inclusions, granules, things like that, warehouses, not factories. So we say they're mainly for storage. They're not containers of complex metabolic systems. Like I said, they have a membrane, but they're not true organelles. One thing I'd like to is give you a little cocktail. So sometimes we'll talk about cocktail knowledge in this class. So there you have a glass with this libation in it. And I'm saying, so when I say cocktail knowledge, these aren't things I'm going to test you on an exam. They're just kind of trivia to know. All right. They're things you can bring up if you're trying to strike up conversation with someone at a cocktail party. So I call it cocktail conversation. And so the cocktail conversation that has to do with inclusion bodies is that some inclusion bodies contain these things called polyhydroxybutyrate right here, and so if I were to kind of marking it here, and these are inclusions, and they're just warehouses for this stuff, and this stuff is used to make like biodegradable plastics. So if you're at a cocktail party and there's plastic cups, you could strike up a conversation by saying, hey, that plastic might have been made by a microbe, and the stuff that was made came from an inclusion body. All right, and there from then you get talking to them, and before you know it, you're married. Okay, so so there's an example of cocktail, and you'll hear more cocktail conversation as it goes on. All right, last thing, so this was the lecture for Monday. So before Wednesday, it would be good before the live lecture, before you come to the live lecture on Wednesday, please go over the asynchronous recorded lecture 2. All right. It says asynchronous recorded lecture 2, and it's kind of longer. So just make it easier to load up, I divide it into 2 parts. It's one of the rare ones. Okay. But again, a lot of it's just review stuff. I think a lot of you will know most of it. It's just a review, but it's good to go over that, listen to that stuff. All right, before you come to the live one on Wednesday, because the live one on Wednesday, I'm gonna again kind of go over some certain lectures points that may have proven to be difficult, and then we're gonna do questions and kind of show you how to do questions on those. So, all righty. So until then, thanks so much. Stay healthy and take care. Bye bye.